Okay, I have the extreme pleasure and honor of introducing the awesome science team, or awesome genetics team of K-State University. First of all, we have Bob Weaber. And Bob, Jackie would like you to go to the back of the room to get mic'd up, if you would. As I was trying to think of what to say to introduce these people, you know, we have a bunch of information in your brochure that you received in your purple folder. So I won't go into, you know, their bio like that or anything. But I realized that I've known Bob for 25 years as of this month. Bob was finishing his beef industry leadership master's degree under Tom Field at the time when I started my graduate school education in breeding and genetics. And apparently Bob must have thought that breeding and genetics was where it was at, because he came back after working in the industry for a couple of years to get his master's in breeding and genetics. You know, so I've known Bob for a long time. I still think of him as a kid, but, you know, geez, Bob, you have to be in your 40s now, huh? And you had hair back then, too. Okay, then, next person I met was Jenny Bormann. Jenny, I don't know, oh, Jenny's in the back of the room. Jenny was shopping for grad schools back in '97, and I met her when she came to CSU. And she was just fresh out of getting her bachelor's at Iowa State. And I've come to know Jenny over the years as well. That was 22 years ago when I first met her. Jenny is an avid horsewoman as well. She's from Iowa. She loves the quarter horse industry, and she's one of the few beef cattle geneticists that I know that is interested in equine genetics beyond coat color. Okay. And then there's Megan Rolf. Megan, where are you? Oh, she's sitting, I couldn't see you. Hi, Megan. Okay, Megan, before I worked at Simmental, when I got out of school, I went directly to the North American Limousine Foundation. I was 41 years old, and Megan was in high school showing limousine cattle. And I'm really excited to know that Megan decided to go into breeding and genetics, and she actually went into the molecular side, which is neat. So all three of these people just have, you know, I've known them for so long, I still think of them as so young, but here they are, you know, in their prime, you know, still working in beef cattle genetics. So, you know, they're awesome scientists, all of them. They all have, you know, you can read their bios in your brochure, and I'm just very pleased and honored to have known all three of them in my career. So I'd like to first, well, Bob, why don't you come up, because they're going to kind of tag team their presentation this morning for you.
Super. Thanks, Lauren.
Sure, Bob.
That was a little kind of... we haven't known each other for a long time.
I know,
it's hard to believe it's 25 years.
I know.
Or
Can
more.
you believe
I know,
that?
but
yeah.
It's been good.
Yeah.
So thank you for that nice introduction. Time marches on, right? And that's been fun to watch young people grow. So I'm closer to Lauren's age than many of you probably realize, but the young folks that come in our business are really important. And so we're, well, little brag. So Megan, who's on the program later, was my second master's student at Mizzou. And if all graduate students were cut from the same cloth she is, life as a professor would be just peachy, right? She's been really good, and I'm glad to have her as a colleague, as well as Dr. Bormann. Jackie was a student at Mizzou when I was there, and so it's been fun. And of course, I've known many of you in the room for a long time. When I was a grad student at Cornell work closely with Simmental, and Jerry Lipsy and Marty Gropp are close friends of mine, and that's how I got to know many of you. So it's a real pleasure to be here and share some thoughts with you. I think I was actually at the first Fall Focus in Bozeman. I think so, and it's always a joy to see a group of progressive seed stock and commercial producers come together and be engaged with the leadership. And as a former Breed Association staff member, there was nothing more frustrating than membership that would complain about the direction of the organization but weren't involved in forming the direction of the organization, right? So if you don't vote, you can't bitch, right? So that's the rule. So you all are here, so I'm preaching to the choir a bit, but that engagement is really critical and really important. So I'm pleased to see this tradition of Fall Focus continue at American Simmental. With that, again, welcome on behalf of our faculty and staff. We're certainly pleased you chose Manhattan as a place to come. We think it's a fantastic place, and so we're glad to share that with you and appreciate you being here. Again, Mike, welcome. This is, I think, maybe the first. We've been kind of going different directions for the last couple of weeks, and so it's nice to see Mike here. And of course, work closely with Ken over time, and so Ken, thanks again for all your service to our department and over time. We appreciate that very, very much. So I want to get into a little bit of, as Jackie mentioned and Lauren, we're going to kind of tag team our talks a little bit. So we'll have the three of us kind of rotate through some of the topics, but the first one that was on the docket is a discussion of the genetics of fertility. And this is one that, you know, as a kind of a cow-calf guy at heart, that strikes pretty close to my camp, and a thing that I think is really important for us as seed stock producers, but I think ultimately one that's very, very challenging. And as we think about evaluation of genetics of fertility, there's a lot of things to think about. And so we'll talk through those. We'll kind of give an overview of the current genetic evaluations across the various breeder organizations here in the U.S. and a little bit internationally, I guess. What's in a phenotype? So we're going to talk through, we say, well, we'd like to improve fertility. Well, what the heck does that mean? When you start thinking about how to describe the reproductive attributes of a set of breeding cows, there's a lot of things to talk about, okay? And that's a little bit, I'll give a little bit away. That's why we've not made much progress, right? There's a lot of things that influence reproduction in beef cows, and that's a challenge for us. And then I want to talk a little bit about some of the opportunities we have, though, for the future, and in particular, a paper that came out this spring from our colleagues at the Meat Animal Research Center that I think is just, it's one of those papers where you read it and go, dang it. Why didn't I think of that, right? I mean, it's genius kind of work, and it's really simple. And when you see the authors on the paper, you won't be surprised by the good thinking that came out of that particular study. Okay, so what is the ideal beef cow? This is, I think, maybe as we start to understand what it is our cows do, and in particular, thinking about this from the perspective of our commercial clients, what makes a great beef cow? Right? There's lots and lots of definitions, but I think the sort of cow-calf guy in me thinks about, well, these cows' job really is to go out and convert sunshine and green grass and that into edible, high-quality beef products for consumers here and around the world. Okay, that's ultimately what they're supposed to do. There's a lot of pieces to that. But we like them to have minimized maintenance costs, so we can talk about, it'd be nice if this was actually a profitable enterprise, right? Make some money. We need to have a cow that produces enough, is maternal enough to produce enough milk to raise a good calf. Down here, maybe should be at the top of the list, gets pregnant on time every time, so this idea of having a calf every year is central to the idea of cow herd reproduction, but also core to cow herd profitability. So that sort of, I think, maybe sets the context for, gets us thinking about, well, what attributes go into that are really important, okay? A little bit of discussion about current evaluations, so ways that we have tried to describe reproductive performance or fertility in beef cows. Of course, American Angus publishes a heifer pregnancy EPD. Brangus has age at first calving, heifer preg, stayability. So there's kind of some variety through here as you read through the list. Stayability is probably one of the more widely adopted concepts. And so that's the idea that, given a cow had a calf at 2 years age, what's the probability she has a calf record, a weaning calf record at 6? Some permutations on that to capture information about cows at younger age points. So, if a cow fails as a 2-year-old, what's the probability she's got a calf at 6? 0, right? She got culled and she doesn't make any more records. So early cows or early failures we can capture information from, and a number of breed groups have worked to try and capture that information. My question to you, does this capture everything that's important about cow herd fertility? I don't think so, and I don't think so by a stretch. It captures some of the things that we can get out of the data we've got, but I think there's more opportunity for us moving forward. Okay? So that's a little bit of the transition here to what is in a phenotype. So I think it's really important as an animal breeder that we sort of precisely define what it is we want to improve, right? So we know that if we've got really a broad description of the phenotype, our ability to really control the underlying components of that, if there's a lot of underlying genetic variants to it that are maybe not all pointed the same direction, so negative or positive genetic correlations, those phenotypes become really, really difficult to analyze, okay, without a lot of work in terms of correlated trait stuff. So it's important we define what it is that we're after. So we can think about things that, if we sit down and think about beef production, well, what attributes or what points in time might we measure something that's important to cow herd productivity? Certainly pregnancy rate is one we think about and encourage cow-calf producers, preg check cows at the fall, kind of see what we've done, both as a reflection of reproductive performance, but also a reflection of management and potential management changes we may need to make in our production system. Rebreeding, so the idea, let's think about maybe a 2-year-old, okay, she has her first calf. We know one of the big failure points of beef cows is that transition from a 2-year-old cow to a 3-year-old cow, so getting cows rebred. The breed association culling data bears this out, that's the big hurdle for most females, is that rebreeding, so we can talk about that as a trait. Fertility. Dr. Odie, how would you define fertility? Ability to get pregnant, right? And so that, I think, we sort of throw fertility around, but we can actually tie a very specific description to fertility, and that is, did she conceive? Okay? Well, that's easy to describe. Is that easy to measure? Okay? The flub in that is if we use a calf record, right, did she calve? Well, there's 280 some plus days in between when the calf was conceived that she may have lost that conceptus. So it becomes a little bit of a messy phenotype as well, because we have early embryonic loss that may be from a variety of factors that we don't capture in terms of data points. Okay? Cow longevity, if we think about our commercial cow-calf producers, culling of young cows, obviously very expensive because of the replacement rate associated with them. So improving longevity or making sure cows make it to aged endpoints has a strong association with cow herd profitability. Okay, days to calving, so we can think about the association of where calves land in the calving distribution. Okay, what do we know about calves that are born at the beginning of the calving season relative to their weaning weights? Okay, one of the biggest influencers of calf weaning weight is what? How old they are. Okay, if we think about a 5-weight calf, okay, that weighs, to make the math easy so I can keep it straight in my head. Okay, let's say they weighed 100 pounds at weaning, they weigh 500 pounds 100 pounds at birth, and they weigh 500 pounds at weaning, that's 400 pounds of gain over roughly 200 days. How much do those calves gain a day? 2 pounds a day, okay? Age is a really important component to describing the productivity of this cow herd, but it's also a reflection of the reproductive performance of the cows, right? Because what do we know about calves that were born early in the calving drop? What did their cows do? Their dams. They bred early in the breeding season, right? So we talk a lot about, in cow-calf production, calving distribution and having that front-loaded. It improves, obviously, calf performance, but also nutrient management, all kinds of things in the cow herd. So that's an important one. So those are connected days to calving, postpartum interval. Again, though, things that are a little bit challenging potentially to measure for us. Okay. Why haven't we made a lot of progress? Well, I gave away part of it. It's stinking complicated, right? There's lots of factors that affect reproduction in cows. Okay, conception and is the result of a whole series of stochastic biological processes. Okay, that's science speak for there's lots of variation from lots of inputs that affect conception, right? And conception is kind of a binary thing, right? I don't know if you remember at least I remember as a young person, my parents saying there's no such thing as half pregnant, okay? The cows are either pregnant or they're not, okay? 0-1 outcome, okay? What about all the underlying things that go into that? Some of those are binary in terms of influence, but most of them are a range of either management level or nutrients or various other managerial inputs that affect that binary outcome. And so it's pretty hard to sort of capture that without a lot of production data, okay? So environment, so temperature, feed availability, bug or pest load, obviously genetics, and in particular genetics with other traits, right? There's some things in our production system that are valuable in terms of calf productivity that might be antagonistic to how our cows perform reproductively, okay? What are the two big ones? Milk and cow size, right? So the two things that affect maintenance costs and input costs the most have a big influence on reproductive performance, okay? Other things, though, that help us, like heterosis and crossbreeding, certainly have positive association with reproductive performance and longevity. Certainly recessive conditions, so if we think about early embryonic loss, one of the main sources of that can be genetic defects or recessive conditions, okay? Obviously management, okay, so things like what kind of breeding system do we have in place? Do we abnormal or imply or enforce a very short breeding season, right? So sometimes we create open cows by not leaving bulls out long enough, given the calving distribution that we just went through, okay? So management plays a role. Obviously bull fertility, exogenous hormone treatment or synchronization programs. We can induce cows to cycle and conceive a calf that ordinarily may have a much longer postpartum interval if we don't put some progestin in them to get them to cycle. Okay? So lots of things that create variation that in most of the data that we get at breed associations, we don't capture any of that stuff, right? So we have to be very cognizant of what it is we do. I think this is one of my favorite, as an extension educator, one of my favorite depictions of how a cow works, okay? And I put this on here to primarily point out that the thing that we're most interested in many cases, is the first thing that goes, okay? So this picture came from a Short and Adams publication in the late 80s. I think, if I remember right, Dr. Odie, Bob Taylor had this in our beef production class at CSU. And it really resonated with me because it describes the nutrient inflow to the cow over here in a variety of sort of nutrient densities or energy densities from, a high concentrate, sort of, you think corn-based diet or a roughage-based diet. And those nutrients go in the cow, and this up here is the reservoir of surplus energy. So that's body condition score in the beef cow. Down here we've got spots where those nutrients go, and so it starts filling up this tank down here first, right? It's the bottom, it's the lowest spot, so basal metabolism gets filled first, and then things like growth and basic body mass maintenance, so maintenance cost of the cow goes here. And then as we improve or have enough nutrients to fill that tank, we start filling this tank. But there's a set of valves back here at the back that get turned on or off to adjust how much nutrient reserves there are, or are a reflection of their performance of those nutrient reserves. And so, if we think about cows on a roughage-based diet, body condition score 4 to 5 is kind of where they live, okay? We dip into the 4s, we start turning off valves, right? What's the first valve? Estrus cyclicity, right? What's the most important thing cows need to do to get pregnant? They got to cycle, right? So if we drop nutrient availability substantially in the environment, we start turning things off, and other things go, right? Lactation, pregnancy, so the cow tries to allocate nutrients to support the calf that's at side before she'll make another one, right? Or maintain a pregnancy ahead of lactation. So I think this tells a lot about cow management, okay, and how important the management side is, but it influences the phenotype, right? And the phenotype is an expression or a combination of genetics and environment. We can't think about fertility without understanding the various influences of environment, okay? Another thing to think about, though, is how different are seed stock production practices from commercial production practices, right? One of our jobs as seed stock breeders is to create new genetic combinations, right? Science speak, we can talk about meiotic events, right? So combining segregation events of sperm and egg from a genetics perspective, putting those together and figuring out, do we make a good combination by chance or not, okay? One of the key drivers in how much genetic progress we make, you guys remember the breeder's equation, right? What's at the bottom? It's a reflection of time. It's generation interval, right? One of the ways to speed up progress is capture more meiotic events, right? More matings, okay? So we have a tendency in the seed stock business to want to shorten generation interval, okay? Does that enable us to evaluate the longevity of the cows in our production system very effectively? I'd argue no, and maybe in some cases, depending on your breeding system and how you use AI and embryo transfer, it might not provide very good information at all about cow longevity, okay? That's almost 180 degrees from what our commercial producers are doing, right? Their goal is to keep cows around as long as they can because they represent, as replacements, the biggest cost center in cow-calf production. Everybody's nodding, right? We see the problem, right? So we've got to have ways to capture information. Okay, and I think one of those is, well, how do we align with commercial producers that are using our genetics, either as breeders or breed organizations, and pull that information into our genetic evaluation system? So how did the yearling bulls we sold 10 years ago look in terms of their daughter's performance, in terms of reproductive fertility, longevity, okay? I don't know another way to capture the data, right? There's some other things we can talk about, and we will in just a minute, but if we really want to understand how our genetics work in our customers' herds, well, we got to go talk to our customers, right? And when I say talk, I mean collect data, right? I'm a scientist. You know. In God we trust, all others bring data, okay? Bring the data. Okay, so data recording, Simmental and a number of other breed associations, Red Angus was a pioneer in this area of whole herd reporting, right? So how can we capture the data, particularly for larger or western range environments that use a lot of natural service sires? We can capture some good information from seed stock herds, but we need some data, right? So we need exposure date data, so the beginning of the breeding season. We need some bull cohort information. We need to be able to understand the breeding system, so if there was synchronization or AI or AT in that, we need to be able to account for that, okay? Another perspective, though, is how do we use the data we've already got, okay? Because there's some actual really interesting opportunities, particularly when collected under whole herd reporting, that provide data to us, right? A calf record, so a birth record, is evidence of reproductive performance, right? We know that cow conceived roughly 280 some days before that, okay? What we don't know is, well, what happened between that point and her previous calf record? How many exposures or how many matings did it take to produce a calf? Maybe useful information. Did that female have exogenous hormone treatment? So is she synchronized? We got to be able to account for some of that. But a calf record obviously provides some good information. Disposal code data. I've had opportunity recently to look at some disposal code data from a couple of breed organizations, and one of those I worked at, it was the Gelbvieh Association, okay? Some of the data was really good. You could go through individual herds and look and see people were really diligent about recording what appeared to be accurate disposal code data, why the cow left the herd, okay? Was it a reproductive failure? Did she break a leg? Did she have a bat? Whatever, okay? There's other data reported because it's under whole herd reporting, right? And so you get billed, you get an inventory-based fee charged against your account if you leave those cows in. So people like to call the cows off, right? It decreases their bill with the association. What do you suppose some breeders do? It's like code 89 or something. Other, right? All the recordings for disposals are others. Suppose that's real? Or is that lazy? It's lazy, right? So this is a plea to you, okay? Be accurate in your cow disposal data, because at some point in time, some grad student somewhere is going to look at it and they're going to go, geez, I don't understand this. And they're going to go to their professor and go, what is this? And then you got to explain to them, well, this is just crappy data, okay? So be diligent in that, because there's really useful information in there, okay? Commercial herd data, we already talked about this a little bit. Obviously, genomics over time, this is painful and expensive data to collect, okay? Genomics allows us the opportunity to leverage that investment either individually or organizationally to capture that genetic variation, okay? So I'm gonna plead to you, good disposal data is really helpful to us, okay? What should we measure? Okay, we've talked a little bit about what kind of some of the different traits are and the environmental influences. I did a little bit of an ad hoc survey, actually right before the BIF meeting this year, and had a good conversation with Mark Thalman at the Meat Animal Research Center. I asked him, well, Mark, what do you think we should measure? And I was expecting those of you that know Mark, once in a while he can give kind of a protracted answer, right? He used 3 words: fertility and longevity. And the conversation was pretty much, that was it, right? We talked about some ideas around that, but he's really convinced these are the 2, and Mark's one of the great thinkers in our genetics world on the beef side. Some challenges in there. Fertility is really a 0-1 sort of conception indicator. To infer breeding season is important in that, or have breeding season data. So bull cohort, turnout date. So there's some additional data points we have to capture with that, which means as breeders, that's more stuff we need to send. And we're sensitive to how much workload we put on you. And so that's, I think, an important trait, but one that maybe is a little higher hanging fruit. Days to calving, okay? So if we know there's a group of cows that are naturally exposed, so don't have AI or ET or synchronization protocol applied to them. This one can be very informative about that postpartum interval and how quickly those cows come back to cyclicity and breeding, okay? Longevity, okay? So good whole herd reporting where we've got information about calf births and production records can be very useful. And those oftentimes don't really require we do much besides just whole herd report, right? So send in good birth information. If the cow calved and the calf was dead, she still conceived, right? There's reproductive information in that. So that can be very, very important. And we're, I think, across many breed groups working to capture and utilize that kind of data. There's been a big push, particularly in the Southern Hemisphere, to sort of get around the idea of days to calving or postpartum interval. And so there's a variety of evaluations. This one, I think, some details from Australia, but really looking to understand the influence of particularly this period of postpartum interval. If we think about gestation length from a fertility standpoint, that's more or less kind of a fixed period of time. We know it varies a little bit, but by and large pretty constant relative to the variation we observe in postpartum interval. So details then on all the calves born, dead or alive, are important. But this helps us understand that sort of performance of cows. Did they rebreed in a timely fashion in the calving distribution? Which I like. I think that's important. But there's still a really valuable bit that we don't capture about the reproductive performance of the cows, right? Is a cow that calves the last day of a 90-day calving season as valuable to you either in terms of calf weight or fertility, reproductive performance, as one that calves on the first day. I think there's a big difference, right? There's a big difference, particularly in subsequent risk for reproductive failure, right? The cow that calves the last day of the breeding season, if you run a fixed, or calves the last day of the calving season, the subsequent breeding season, how many opportunities does she have to conceive a calf? Probably one, okay? And on average, Dr. Dykman, what's the reproductive success of a single mating in terms of probability? It's a little better than 50-50 things, hopefully, Driggers, right? It's about 65%, right? So a 35% chance that this isn't going to work out, and what happens then? She's open, off to town, okay? She's a bad bet, okay? So the cow that calves, or the heifer that calves at the beginning of the breeding season, a much better bet, okay? I like to leave, I don't go to Vegas very often, but Vegas is kind of a fun spot. Put all the gambling in Vegas, right? Don't do that with cows that calved the last day of the calving distribution. It's a bad bet, okay? It's a bad bet. So what other things might we do? Well, this is where the genius I talked about earlier. If we think about the performance of cows in our production system, those cows that calve early in the calving season, reflected they bred early in the breeding season, those have a lot of value to us. How might we use that information in a genetic evaluation? Well, Mark Thalman, Larry Keene, Warren Snelling, Bruce Golden, I'm trying to think, there was 5 authors on that paper. I'm leaving somebody out. But Warren was the lead author on this paper. And what he looked at was really taking existing data, and they used the Meat Animal Research Center data, but we could extract this from any of the IGS partner databases, and looked at principally two measures of cow herd cumulative productivity. One of those was just the sum of the age of the calves from a particular cow during her productive life, okay? So we know remember the oldest calves are the most valuable ones in terms of thinking about where they landed in the calf drop and their expected weaning weights, okay? So this sum is really useful because it tallies up, a cow that has a calf at the beginning of every breeding season, she's got the biggest number, okay? What happens to a cow that misses a calf? She gets a 0 added that year, okay? Go to the next one, okay? So it spreads them out. It points, then we sort of look at, well, what's the biggest number? That's the easy thing to do, right? And it points us the right way. The other one is cumulative pounds of calf weaned in terms of actual performance, not adjusted records, but actual performance, okay? It's kind of a reflection of the same thing, right? Those cows that calved early, their calves on average are going to be heavier at the weaning date. So kind of everybody's going, oh yeah. I'm like, I read this paper, I'm like, well, what the heck didn't I think of that? Okay, it's a pretty tractable way to measure reproductive performance. And this sort of cumulative age or cumulative weight, this was a JAS paper that came out earlier in 2019, looks to be a really useful measure, okay? Here's this is the heritability estimate, so they evaluated a lot of other traits in there. Pregnancies, number of calves born, calves weaned, so a tally of the count of calves weaned over a cow's productive life, weaning age, weight weaned, and then the association with cow body weight, okay? The interesting thing is as we think about fertility, most of us have in our head, well, the heritability of fertility traits is low. You get out here to measuring cumulative cow weaning weight to 8 years of age, okay? Or calf weaning weight, okay? That's this blue line. No, sorry. That's this blue line. Dang, the heritability is 0.4. I'm a lefty, and so I like the picture. So when I pulled up this paper and I started looking at it, I started looking for the plots, right? 0.4 is awesome. What other traits have heritability of 0.4? Yearling weight, it's close. It's in the upper 0.3 somewhere. Marbling EPD, how much progress have we made in yearling weight and marbling over time? A bunch, okay? What might we do if we implemented something like this, okay? Pretty useful. I'm going to skip over that. The 2 key points are the correlations year to year are really high, okay? So if you go, what's the weight weaned from a 2-year-old to a 3-year-old, those correlations are really high, okay? But as we start thinking about, well, what if we spread out, if we use 2-year-olds as indicator data for, say, productivity at 8, okay? Those correlations go down, but they're still very strong. So the data we collect in young cows provides us good indicator data of later productivity. Okay, and that's sort of bears out in this set of plots. And I put this mostly as a reminder to tell you that for the traits of cumulative calf weight and age, the correlations go all like, they match up very closely to what we think about, okay? So what do you expect happens if we have cows that are very heavy in terms of body weight over time? Are they more or less reproductively efficient than cows of lighter body weights? They're typically less, okay, and that shows up in this data, okay? So all the common sense things sort of check out, which is again kind of the, this is potentially a pretty useful tool, okay? So I'll leave that with you for kind of food for thought. I'm really pleased to be here. I'm going to stick around for the whole day, so I look forward to having some good conversations with, I know Chuck's already got questions for me. Chuck Miller, good to see you, buddy. So I look forward to interactions with you through the day, and again, appreciate the opportunity to be here. Thanks. So our next speaker will be Dr. Megan Rolf, and Megan is going to, I'm trying to remember our topics. We divided these up a while ago. And
bull fertility.
Bull fertility, the male side of this equation, right.
Thanks, Weaver. I had that problem earlier. I should be open down here. Okay, well, thank you all very much for the opportunity to be here today and to visit with you a little bit about a project that we're just really getting started at K-State. But it's one that I'm pretty excited about and one that I've been excited about for quite some time. We've been looking for data. But when we think about male fertility, when we think about fertility, we typically think about fertility from the female side, and that's a lot of what Bob talked about this morning. But I think what we need to make sure that we don't forget is that a significant percentage of reproductive failure is failure that we can actually attribute back to the male, right, to the bull. And that's usually due to either poor semen quantity or subfertility due to poor semen quality, and sometimes the health status of the animal as well. But when we think about the probability of a cow conceiving, it's really contingent on the female as well as the male. And I think sometimes we tend to forget about the male in that equation. So the bottom line is that the bull does contribute substantially to the reproductive success or failure of the cow herd, and we all know that that is a really economically relevant trait for cattle producers. When we have reproductive failure, we can get longer calving seasons due to delayed conception, so we can get lower weaning weights, and that's something Bob talked about quite a bit. We can increase our culling rates and maybe erroneously cull cows when it really wasn't their fault that they didn't get pregnant, right? It might be the bull's fault that they didn't get pregnant. And one thing that I think we sometimes forget about as well is that when we have repeated breedings, we also up our risk of injury for both the cow and the bull. So I would argue this is a really, really important trait that we've sort of forgotten about the past few years. And there's not a lot of data out there that really compares male fertility and female fertility and tries to integrate those two concepts. There are a few studies from dairy cattle in around about 1980 that look at the genetic correlation between male and female fertility. And in those dairy populations, it looks like it's probably positive but modest. So as we increase male fertility, we would also increase female fertility. The only studies that are available in beef cattle are kind of between scrotal circumference and days to calving in Hereford Angus and Zebu crosses. And those are actually negative. So as scrotal circumference would increase, days to calving would decrease, okay? But that's a favorable correlation. So it looks like the majority of the genes that create variation in male fertility have male-specific functions, okay? Because those correlations are low. So selection to improve female fertility should result in a positive influence on male fertility, but it's really not optimal, right? Because a lot of those genes that are contributing to male fertility appear to be unique and specific to male fertility function. So if we really want to improve overall efficiency of beef production, we really need to not only focus on female fertility, but also not forget about the male side of that equation and try and develop tools that will let us eliminate young bulls with sperm abnormalities and unacceptable semen quality earlier in their lifetimes before we've invested so much time in bull and money in bull development. So we have, of course, breeding soundness examinations, which primarily function as a culling tool in the industry. But the big challenge I see with using breeding soundness examinations as a culling tool is that we've already invested a lot of money in developing those bulls, right? So when we've already invested that money and then we have to cull that bull at a year or a year plus of age, we've basically lost that investment, right? We're left with the salvage value of that animal. And really, we don't have any genetic selection tools in the industry at this point in time that really address that need. We have scrotal circumference, but in scrotal circumference is pretty highly heritable, as Bob said earlier. When we have a heritability of 0.4, we can make progress, right? But the point I want to make with this table here is that we have a lot of other traits that we could measure that are related to semen quality and quantity that should affect male fertility that are also pretty highly heritable, right? So these are low to moderately heritable depending on the study. It'll be interesting to see how these hold up when we get really large populations of animals, but it looks like we've got some pretty substantial heritability estimates here. So these are traits that we can make progress with. So I want to take a few minutes to just kind of go through some of the traits that are options available to us and for which we have data on at Kansas State at the moment, and just kind of give you some background on those different traits. So ones that you're probably very familiar with, even from breeding soundness examinations, would be sperm concentration, which a lot of times we're measuring with either a spectrophotometer and light emission, or with this hemocytometer here. We also have volume, which is, of course, how much volume there is in the ejaculate. And if we look at total number of spermatozoa, we can just get that from calculation, right? So if we have the concentration of sperm per milliliter, we know how many milliliters we have. Multiply those together, we can get total number of spermatozoa. So we would expect those to be very, very highly related to the number of spermatozoa. And of course, everybody's going to be familiar with scrotal circumference, right? So we do have scrotal circumference. So here I'm going to have to exit out of the presentation so that we can watch this video. But the first sort of suite of traits that we also have available is related to motility. And motility can be done as a score, which a lot of times you see in a breeding soundness examination. But we can also have motility recorded as a percent. When you start looking at data from bull studs, a lot of times that'll be recorded as a percentage. And if we think about motility, let's just kind of watch this video here.
In this video, we'll be looking at sperm specimens with varying motility.
And you can't see the video. Slide it over? Right or left? Okay. Okay, we'll give that a go, although I can't play it now, so I guess we're not going to watch the video. Never mind, just kidding. Just kidding. But basically, the video just showed different sperm samples. So when you look at those under the microscope and they're moving around a lot, right, that's going to be a high level of motility. And when you look at those samples and there's really just not a lot going on, those are low motility samples. We also have, in addition to motility, we have progressive motility. So it's really important that the sperm not only move, but that they move with purpose, right? So these sperm here that are non-progressively motile are just kind of doing donuts in the uterine parking lot, right? So if they're on a road trip, they're not going anywhere, okay? So we don't want sperm doing donuts in the uterine parking lot, right? We want sperm that are going on a road trip that are headed somewhere with purpose, right? So that's one of the things that we look at with progressive motility. And then, of course, we have percent normal sperm when we start to think about the actual morphology of those sperm. So we can look at sort of these gross measures, like what percentage of them are normal out of the total number in the sample. And if we look at sperm that has been frozen and then thawed, like you'd find at a bull stud, we can look at this percent living. So if you see these sperm here, the ones that are dead sort of take on the stain in that, and they look a lot like the color of the background. Whereas those live sperm, you can see that they are much brighter, and they really stand off of the background of that. So you can figure out what percentage of them are still alive after freezing. Okay, so if we really dig down into abnormalities, sometimes we have specific abnormality data. So one example would be droplets. So percentage of sperm with abnormal cytoplasmic drops. So we may have proximal drops, or we may have more distal drops. A lot of times you'll see that in younger bulls, in samples from bulls that aren't quite as mature. So this plot here shows, on the top part, that's percent normal sperm in the blue. And on the bottom, we have primary and secondary abnormalities. So those drops will be in the secondary abnormalities. And what you can see is as this particular bull got older, the percentage of those secondary abnormalities dropped. And due to the additional information we have on this bull, we can tell that he had a lot of droplets at the beginning. As he aged and matured, the droplets dropped off of that sample. Okay, we can also have head abnormalities. So these can be things like double head, pear-shaped head, or elongated heads, a variety of different head abnormalities. So just looking at the percentage of those in the sample. We can have tail abnormalities, so bent tails, coiled tails. And then we have sort of more gross groupings of those abnormalities. So primary abnormalities will be those usually associated with abnormalities of the head. Secondary abnormalities will be those typically associated with the tail. And then, of course, total percent abnormalities. So we'd have total percent normal and percent abnormal. So the real question is, are these traits necessary? There's a lot of traits we just talked about. Why not just measure scrotal circumference? So scrotal circumference is genetically correlated to the motility of the sample, as well as a variety of the different abnormality percentages and percent normal. You can see that those do have a little bit of a range around them, and the correlations are mostly favorable, but they're mostly also pretty low, right? So we would expect, as we select for increased scrotal circumference, that we would make some progress in these traits, but there's probably going to be a limit on what we can really do without specific tools geared towards these specific measures. So the real question is, which ones of these traits are most related to the conception of the female, right, and male fertility and fertility success? If we look at genetic correlations of the quality traits amongst themselves with what we see in the scientific literature, you can see I've colored the green ones as those that are favorable, the red as unfavorable, and the yellow are things that there's just not a lot of data out there. We might have one estimate or two estimates from one study, but not a lot of data. And so rather than focusing on any specific numbers here, because I know there's a lot, my point here is simply that we don't have a ton of data on a lot of these traits in a lot of different studies. We don't have a lot of replication in a variety of different populations. So I would argue that we have a lot to learn about these traits and their relationships with each other, as well as other economically relevant traits within the industry. So in this project, we have sort of four main goals. The first is to evaluate the impact of male fertility on conception rates and develop a sire conception rate EPD like we have in the dairy industry, but develop that for the beef industry. And the second is to provide sort of a prototype evaluation of male fertility in beef bulls using semen quality metrics that we might collect either from bull studs or for breeding soundness examinations. And so both of those two top goals are really focused around the development of selection tools specific to male fertility traits, other than scrotal circumference. The third here is to identify and validate regions of the genome that have variation that is important for the prediction of genetic merit in male fertility. So basically leverage that genomic information like Bob talked about for female fertility, but to do that from the male side of the equation. And finally, we want to look at the genetic relationships between not only the semen quality traits amongst themselves and scrotal circumference, but also with other economically relevant traits within the industry. So we really understand the impact of selection on these traits and what that will do for the bottom line. So we've been working to gather a variety of different data sets, and I'm going to show you quite a bit of data today and not a lot of analysis because we're just getting started here. But we do have 3 primary sets of data that we're working to gather. The first is conception data. So a lot of what we have committed to the project thus far is beef on dairy conception data. So a good chunk of that is coming from DRINC, the Dairy Records Intelligence Network. So this morning, Dr. Dykman mentioned a lot of dairies in western Kansas. So a lot of them have signed on to be part of DRINC. And so they have been reporting data on their matings. And so there are 107 cows approximately in that database. And the majority of those matings, which has been over 250,000 in 2018 alone. And the majority of those matings for the past couple of years have been using beef bulls. So there's a lot of data there that we may be able to glean to look at conception. So we're working to establish relationships with other data providers that could give us more beef on dairy data, conception data, as well as partnering with some larger heifer development organizations and, of course, breeders such as yourselves that may have large contemporary groups of beef on beef conception data to contribute. So if you have interest in that, I'd encourage you to visit with Jackie. We do have some tools set up to start collecting that data if that's something you want to participate in. So today I'm going to spend the majority of the time I have left talking a little bit more about some of the data that we've been able to gather from some bull stud partners. So we've been very, very lucky to be able to work with CABSU here at K-State, so Kansas Artificial Breeding Service Unit, and Origin, who've provided a variety of collection data and quite a large volume of collection data for us to work with. So we're very excited to be partnering with them on this project. And so I'm just going to go through a little bit of the data from these 2 bull studs. I'm not going to tell you which one is which. And just kind of look at what that data is telling us, and just in a very high level sort of way before we get started with a lot of the analyses. So this table here shows years. So we've got records from 2004 up to 2018. And this bull jumps column basically gives you the number of collection records. So about 67,000 different collections throughout that time period. And the quality control column are the ones that made it through that freezing process, okay, that were high enough quality to freeze. So right off the bat, if I do a little back of the envelope math, we can see that about 41% of collections did not meet basic quality thresholds for freezing. 41% of those samples were lost. So I think we probably have some room for improvement. If we look at the age distribution of those 67,000 collection records, obviously we do not have 67,000 bulls represented in this data set, so some of these bulls are being collected multiple times, right, at different ages. So we have that here. You can see most of the bulls are fairly young when they're being collected. We do have a few bulls of a substantial age in that data set. But I think one of the interesting things about this distribution of ages here is that with these repeated collections, one of the things we may be able to look at is which bulls are stable breeders through a variety, or stable quality semen producers through a variety of different environmental conditions. Because if we look at a lot of different traits, what you see is that some bulls are pretty stable in terms of their performance across environments in different temperature and humidity conditions. And some bulls tend to have rapid declines in their production when they encounter more challenging environmental conditions. So with these repeated collections over time, one of the things we should be able to look at is which ones of those bulls are stable performers. Now that would be an advantage to bull studs because they can collect those bulls over the summertime as well, but I think that may be of a lot of interest even for people using AI sires to produce natural service sires, right? Because a lot of our spring breedings are during a time of the year when it's pretty hot, right? So this could be a big advantage. So this is very, very raw data, okay? These are just moving averages over time. We did not adjust for age or anything else, but I just wanted to give you a snapshot of 5 different bulls that we've got here. So we've got a blue, green, yellow, gray, and an orange line. And really my point here is that we've plotted out moving averages of motility in this first plot here on the left. And what you see is that some of those bulls are pretty stable, right? Those lines fluctuate. They all fluctuate, but some of them are pretty flat. And then we have other bulls like this bull in the green here. he's jumping all over the place, right? He's got samples that for a while are up around 90%, and then for a while they're really down around, 55%, right? And jumps around a lot. So not a lot of consistency there. So we have some that are more consistent, some that are less consistent in motility. If we look at percent normal in this center graph, we see the same thing. Some of the bulls are fairly consistent. A lot of them, including that green one again, jump around quite a bit. And this is kind of interesting because this percent normal in this particular data was collected after thawing, okay? So what this means is we still see that variability in the samples even when they already passed a pretty strict quality control threshold, okay? And then this last one, we just went through and said, okay, if the sample was frozen, it gets a 1. If it didn't freeze, it gets a 0. So sort of what Bob was getting at with the binaries, that we either had success or failure. And then we looked at moving averages of those collections over time. And what we see is that down here, there's a gray line down here. None of those bulls' collections were good enough to freeze. And that's why you don't see them in any of the other plots, right? There were no motile sperm, so it wasn't on that plot. And of course, if there's no motile sperm, we didn't bother with freezing it, right? So he's missing from that plot as well. And you can contrast that bull with the bull in the orange up here, where every single collection was good enough to freeze, okay? And then we have those other bulls that sort of bounce around in between. Some of the collections were good enough, some of them weren't, okay? So a lot of variation in the performance that we see in terms of quality for these bulls. So here we have the number of records in stud 1, and there's a ton of numbers in this table. The only thing I really want to emphasize is that we've got about 3,000 records in that data set with about 67,000 collections on those 3,000 bulls. For the Simmental specific data, we do have 350 animals in there and about 7,000 collection records. So I thought I'd just put up some summary statistics to kind of give you an idea what data exists in those data sets and kind of what that looks like. We're not going to go through all of it because there's a ton of stuff here, but these 3 right here that I've boxed up at the top have to do with motility. So the first one is pre-freeze motility. The second one is post-thaw motility. And then the third one is motility after 3 hours, after it's been thawed and then sat there for 3 hours. And the means are kind of interesting. Obviously, they drop over time. So we start at about an average of 50%, and then it drops after freezing to about 44%, and then after 3 hours, dropping down to about 13%. But if we take a look at the minimums and maximums, what we see is that motility is ranging in these samples anywhere from absolutely none up to 100% motile sperm, okay? And even after freezing, still going from 0, sometimes the freeze process, these are the ones that pass quality control. So sometimes the freeze process will result in 0 being motile after that, up to about 75%. And then 3 hours post-thaw, we saw the mean drop to 13%, right? But we still have some bulls that have 60% motility after 3 hours. So I think that's kind of interesting. Now, if we take a drop down and look at the abnormalities, we can see running about 11 to 13% for primary and secondary abnormalities, about 75% normal sperm. But again, we have a really large range in those values. So ranging from 0 to 8 on the low end, all the way up to about 65 to 100% on the top end. Finally, scrotal circumference in this data, just to give you a frame of reference here, the mean was about 40 centimeters, and then the min would be 21, and the maximum up to 55 centimeters. Okay, if we take a look at the records from stud 2, we've got about 900 additional records and about 900 additional bulls and about 14,000 additional collections. For Simmentals, that's adding another 100 animals and about 1,400 collection records. And if we take a look at the summary statistics here, we can see there's some different traits being recorded at this second stud, okay? So combining these is one of the things that we still need to do some work on. Again, I'm highlighting the motility here. This again is post-thaw motility, and then 2-hour motility are the ones in the box here. You can see it's pretty comparable to this pattern that we saw in the first stud. And again, a lot of variation with the minimum and maximum values there. One of the things percent normal here, as was in the other data set as well. about 73% was the average. I think it was 75% in the other data set. So again, pretty comparable. Minimum ranging from 0 up to a maximum of 97%. What I think is interesting in this data set is the abnormality data. So these were not aggregated into percent primary and percent secondary abnormalities in this particular data set. So we do have some records here with specific abnormality information on heads, tails, droplets, et cetera. Although we can group those into primary and secondary to combine them with the other data set when we go to analyze that. So the final data set that I'm going to talk about today is breeding soundness examination data. And this is where we're really reaching out to all of you. We've partnered with a lot of breed associations, but really the ability to collect that data is up to all of you. So if you have interest in male fertility or think it's an important trait, I'd really, really encourage you to think about submitting some data to the project. And if you have interest in doing that, visit with Jackie. She's more than happy to work with you in getting that done. So this is an example of the spreadsheet. If you do decide you want to submit some BSC data, this is the spreadsheet that we put together. So you notice there's a little bit of required data. It's basically who is your bull and when did you collect the data so that we know his age when that data was collected. The rest is stuff that's preferred or optional, right? Because we realize that you will have different metrics depending on where you did your BSCs. So preferably we'd like to get sort of some information on who did that, what the scrotal circumference is, motility either as a percentage or a score, volume and concentration information, and if you have information on abnormalities, we'd love to get that as well. So I'd really encourage you, if you think this is an interesting trait, to consider submitting some data. And to provide an incentive, we do have a few large data sets from breeders, so I thought I'd give you just a little peek at a few of them. Again, not identifying the breeder, but for breeder 1 here, to give you an idea of the different types of data that we've had submitted, this particular breeder has motility score that ranges from basically none up to very good. You can see most of the bulls fall in that good to very good range, but we do have some on the lower end. So the primary traits that were submitted for this particular breeder were scrotal circumference, percent normal, and then percentage of primary and secondary abnormalities. So if we look at the distribution of scrotal circumference, most of these bulls were at about 34 to 36 centimeters. The average was 35. Again, pretty normal distribution of those records. Percent normal was, most common value was between 60 and 70% normal sperm. And primary abnormalities had very few in this particular set of bulls. And secondary abnormalities inched up a little bit from where the primaries were, but most of those bulls were between 10 and 20% secondary abnormalities. Okay, if we take a look at a second breeder here, this breeder had motility on a 1 to 10 scale. So 1 being poor, 10 being excellent motility. And you can see there's quite a bit of distribution here. If they also provided some data on scrotal circumference as well as percent normal sperm. So we didn't have abnormality data from this breeder. That's okay. We can see that the scrotal circumference distribution looks pretty similar to the one before, except we have a few more that were really on the high end of that scale. But again, the average is about 35, and most of the records fall between 34 and 36. And for percent normal, we have a little bit different looking distribution here. So we have quite a few bulls that tended to be on the upper end of that scale with very, very good values for percent normal sperm. And then we had some bulls, kind of a bimodal distribution. We have a second peak there of bulls that had percent normal sperm between about 30 and 40%. So really interesting, I think. And then finally, breeder 3. This breeder submitted the largest number of records. There's about 20,000 BSE records here, but we didn't have data on some of the abnormalities and things like that. So motility, again, on that smaller scale here, you can see most of the bulls tended to be good or very good. Scrotal circumference inched a little higher in this set. 36 was the mean, but again, most of them are between 34 and 36. This data set does have some older bulls in it, so you'll notice the scrotal goes up to well over 46 here on a few of the bulls. And we had percent normal here. The mean was about 81.6. And there again, with the weight distribution, you can see a lot of the bulls were probably yearling bulls, but we had some bulls that were older at weighing in over a ton. And then for percent normal, most of the bulls tended to be on the far right end of that scale, so the good end of the scale, but we did have around about 700 bulls that had percent normal between 0 and 10% in that data set. So a lot of variation that you see. I just did some really preliminary correlations. Again, these are not adjusted for anything, but just to give a rough idea of the correlation between percent normal and scrotal circumference in these different data sets, because those were two traits that they all shared in common. And the correlations for phenotypic correlations range between about 0.1 and 0.3, with most of them falling on the lower end of that scale. So with that, I would be extremely remiss if I did not provide some acknowledgments here. And first of all, Maddie, wave, stand up, raise your hand, something. So Maddie's over here in the red shirt. She's a graduate student in our group. And basically anything you saw that had any data on it today, Maddie did that. So I want to make sure I give her a lot of credit for that. She's a really, really fantastic graduate student in our program, and we're lucky to have her. Also, another graduate student at K-State that's been working a lot with this data is Ashley Hartman. Ashley actually worked at a bull stud for a few years before she came to do her master's here at K-State, so she's been a great resource to talk to you about, sort of collection procedures at studs. Of course, huge thank you to the people who have provided data, so Origin and CABSU, as well as the individual breeders that have signed on to participate in this project. Really appreciative of them. And I want to give a thank you to the ASA staff as well, because they've been really fantastic to work with to get data and start on the analyses of the Simmental data that we do already have. And again, I would encourage you, if you have interest, please visit with Jackie, and we'd be happy to get you a part of the project. And finally, we'd be remiss to not thank our other breed association partners outside of ASA who have contributed to this project as well, and they're listed on the screen here. So with that, I think we're maybe holding questions till the end. So I'll be around through the afternoon here, so I look forward to visiting with you if you have questions or want to talk about the project. And with that, I will invite Jenny Bormann up to the podium. And Jenny is going to be talking to you today about some projects, some work that's been ongoing in feet and leg scoring of cattle.
Is talk a little bit about a project that we've been working on for the past few years. And one thing you may have noticed from the end of Megan's is that if we're doing a good job, we don't do all that much work. We make our graduate students do the work, and then we can take all the credit for that. And so that's a little bit what I'm going to do here now, because most of this work was done by a pair of graduate students at K-State. Lane, who you guys know, Lane Geis, who is now on the ASA staff. And then Brady Jensen, who is now working on a PhD at South Dakota State while he's coaching the judging team there. So I'll stand up here and take the credit, but really those guys did the majority of the work on this. And so this is a study, like I said, we put together a few years ago as really kind of Dr. Weber's ideas. He was out visiting with producers, just some anecdotal evidence that we're seeing more problems, right? We've got some issues with structure, with feet and leg, set with longevity. And so what are some things that we might do to combat that? Could it be that we just haven't paid enough attention? We've selected for lots of other traits, and I don't need to list those, but maybe we haven't paid enough attention to some of what we might call the functional traits. But we also need to think about the fact that perhaps this isn't just a production issue, it's not just a longevity issue, that we don't need lame cattle. That's a problem for everybody, especially when you think about getting to the feed yard and the packing plant. But obviously from the seed stock and commercial side, the cow-calf side, we think about feet and legs, and we think about structure as contributing to longevity, because we don't want to have to keep more replacements, but maybe more on the bull side, right? We've got to have a bull that can stay sound and breed cows, and so something maybe we need to think about spending some time on. Okay, like I said, we know that if cows last longer in the herd, we don't need to keep as many replacements, and I think Bob said earlier today, that's one of the biggest cost drivers for a cow-calf operation, and so if we can keep those cows around longer, excuse me, we're going to save a little bit of money there. But kind of like fertility, feet and leg structure soundness, that's kind of a tricky phenotype. What is the phenotype? What do you measure? What do you score? How do you select on that? And we really don't know what those different traits, what their relationship is to longevity or soundness. But we also know that to make good selection, we need to have an EPD, and so we need to have some sort of numbers to put to this. We need to be able to score something so we can calculate a genetic evaluation, and you guys know that. As with a lot of things, and I think the bull fertility is another example of that, the dairy industry might be a little ahead of us in this area, where they've done a little bit more of this analysis and this data collection. And there's been, over the years, quite a bit of data that shows there's some moderate genetic correlations between what they call type traits, so some of their structure traits, and longevity of those cows in the dairy herd. We know that survivability, stayability, longevity, whatever you want to call it, is overall probably lowly heritable. So maybe if we had some types of soundness traits, structure traits, those might be good indicators to help us predict longevity, especially at an earlier age. If you sort of look at a conglomeration of studies in the dairy industry, you can see that for some of these structure traits, heritabilities are edging up into the moderate range in some cases, and so probably lowly to moderately heritable, but certainly traits that you could make progress on with selection. So currently, the Holstein Association does a genetic evaluation. So this isn't transformative thinking. They've been doing this for a while now. They do a genetic evaluation, calculate PTAs, which are their version of EPDs. They calculate those for lots of different type traits, things like dairy character and other things, but 4 that specifically involve feet and legs. And they're getting, heritability is 0.1 to 0.2%, somewhere in there. And they actually then index those and publish a feet and leg score index to help their producers select for those traits. So very similar to the kinds of things that we do in beef cattle for different sets of traits. But if you go back in the beef industry, Simmental actually started taking a look at this, pushing on 20 years ago now. Graduate student, I believe it was Cornell, is that right, Wade? Is that where Dave was? Collected a lot of records on conformation traits, if you will, and found pretty decent heritabilities for rear leg side view and some pasture and foot angle traits. So not something that's really been big in the beef industry, but there's been some work on that. There was a study in Italy not that long ago that specifically, we looked at a lot of traits, but one of the conclusions they found was that set to the hind leg had a huge impact on how long a cow stayed in the herd. Those posty-legged cattle had 60% higher probability of being cold than sort of that average cow. So some interesting data from Europe. Probably Australia's been a little ahead of us in the beef industry on some of these things. Australian Angus has a pretty big data set where they looked at several different traits, foot angle on the front and the rear, claw shape on the front and the rear, and set to the hind leg, both from the side and the rear. And the purple is the heritabilities of those traits, and getting up there to the moderate heritability, 0.2 to 0.3% in a lot of cases. Genetic correlations on the off diagonals, and you can see that for some of those traits, they're very high. Foot angle on the front and foot angle on the rear, very highly correlated, that would be expected. And some of those others are a little high as well. And that actually led then into their genetic evaluation. So they do have a large-scale national cattle evaluation for soundness traits. And so they calculate EBVs, which is what they do instead of EPDs. But they look at 5 different traits. Front feet claw. Then they look at the pastern or hoof angle, both on the front and the rear feet. And they look at the set to the hind legs from the side and the rear. And this would be sort of the pictures they use to describe their scoring system. And again, they publish that in their regular genetic evaluation. We also know that very recently, just in the last year, the Angus Association has started calculating an EPD for these types of traits. They've decided for now just to do 2. So they look at hoof angle and claw set. Their guidelines say to just pick the worst foot and score that one, so they don't look at all the feet, they score the worst one. And as of this summer, a little bit earlier this summer, they had somewhere in the neighborhood of 17,000 records they had collected and found heritabilities in the 0.2-ish range for both of those traits. And so that's a research run for them at this point, but they're continuing to collect data to continue to try to improve those traits. So what we decided to do with a couple of breed partners, both Red Angus and Simmental, was to try to collect some data on some different structure traits and look at some of these relationships, look at the heritabilities, and figure out if it's something that breeders could use to make selection. So what I'm going to present today is the Red Angus data. Lane is working currently, we've been working on it this week actually, the Simmental data, and so I think that you'll hear about that a little bit later. But we had about 1,800 Red Angus cattle scored, and when I say we scored them, again, I mean the graduate students. They traveled all over the country and scored cattle. And so going into this, we really didn't know what traits to look at, right? There just has not been that much data out there. And so what we decided was to sort of go big and then see where it shook out. So we measured 14 different traits, and we'll go through that kind of quickly here. We looked at body condition score just to have that as some extra data. But one thing that was really cool that the grad students were able to develop was a tablet-based scoring system with these little sliders. And so they would just type in the name of the ID of the calf and then just swipe left or right and slide that slider to what that score was going to be. And so we had for the front feet, hoof angle and hoof depth, the claw shape on that front foot, looking from open divergent to scissor claws. Same thing on the back feet, so hoof angle, heel depth, and claw shape on the hind. One thing we wanted to look at was sort of foot size. You hear a little bit of comments out there about small-footed cattle, and so this was just it wasn't a measurement, it was the score just evaluating, is this a small hoof or a large hoof in relation to the animal? So it wasn't like we were down there with a ruler actually measuring the feet, but it was a subjective measurement. Some front leg traits from the side, buck versus calf kneed, so that front knee and shoulder. Then when you look at them from the front feet, so splay-footed to pigeon-toed, and then knock-kneed or bow-legged. So those two traits were separated. And then the rear leg, this would be similar to the Australian data where you look at the rear leg from the side and from behind. And then the last thing they scored was what we just called composite. So we asked the students, said, okay, if you just had to give this animal an overall score for how sound they are, just give them a number. And so that one was from unsound to very sound. So that one was a little bit different. Okay, so we had every animal scored by at least 2 people, and then those scores were averaged. And then, of course, these are registered cattle, so we had data from the Breed Association. After some data cleanup, there was about 1,700 animals included. And again, we got the pedigree file from Red Angus. So the majority of cattle that were scored were younger cattle, yearlings, 2-year-olds, and a few 3-year-olds. When they went to the farm to score the big groups of yearling cattle, if they had cows up, we scored those as well. So we did get some data on older animals, not a lot, but some. If you look at it, males and females, obviously we were scoring big groups of young bulls, so primarily yearlings and 2-year-olds. And the females, again, primarily big groups of yearling and 2-year-old heifers, but we did get some older cows as well. Real quickly, those scoring systems were 0 to 100, and so of course most traits would be sort of centered around 50. We think of that sort of as being the optimum. But then we went ahead and calculated the heritabilities, and so because it was such a big data, and a large number of variables with 1,500 animals, the guys went through and did bivariate linear models to get heritabilities, and so these would be the average heritabilities from all those 91 different combinations of 2 trait models. And it's encouraging that these are pretty much in line with what the literature would predict they should be. somewhere in the 10 to 20% heritability for most of those traits, although we had a few that were a little bit higher with reasonable standard errors. What's also interesting is some of the correlations, and so if you imagine 14 by 14 correlation matrix, that's a lot of numbers, and so I just tried to pull out the ones that were a little bit more interesting. A lot of them were near zero, right? There were a lot of those traits that weren't correlated with each other, but some of them were. One of the kind of important take-homes, I think, is that if you look at all the numbers except the last one, you can see that those are correlations between front and rear, hoof angle, and heel depth. Well, if we see genetic correlations of 0.85 or higher, what does that tell us? Are those wildly different traits? Or it's pretty much the same trait? Okay, and so what that tells us is that we can probably measure one of those, pick which one, but we're gonna get about the same information as if we measured all of them. So measuring front feet and rear foot, hoof angle, and heel depth might be overkill. We can get enough information from one of those. Similar with claw shape on front and rear, not quite as high, but still a pretty high correlation, and that makes sense, right? If they're bad clawed or good clawed, they're probably the same way on both ends. And so that tells us that we can maybe do some refining of our scale and eliminate some of those traits. One thing that I don't have the number, but the claw shape was not correlated with the angle or depth. So those do appear to be separate traits. Some of the correlations of front feet with leg traits. Turns out that the more upright hoof and the tall heel tends to be associated more with buck-kneed cattle, and more sloped hoof, low-heeled tends to be associated with calf knees, and those were sort of moderate correlations in the 0.45 area. If we look at the correlations from front feet with the rear legs, those tend to be pretty high as well, where more upright hoof, tall-heeled cattle, in that case is associated with being more posty-legged, and the more sloped hoof and low heel is more associated with sickle-hocked. And then if we look at the bottom two, that's front feet with rear leg side view, or rear view, sorry, and in that case, the upright tall heel is more associated with being bow-legged, and the sloped hoof low heel is more associated with being cow-hocked. So what does that mean? That's kind of a good question. We've got some moderate correlations, so could we use leg traits to help us indicate hoof traits or vice versa? That would seem to be reasonable. If we talk about rear feet with legs, that's going to be very similar to what we saw with the front feet, because we know those are very highly correlated, so I won't go through those again. One thing that we did come up interesting here a little bit was that the rear claw shape and front legs were, front knee orientation and hoof orientation tended to be a little bit moderately correlated, so that was kind of interesting. But again, could some of these leg traits help us indicate foot traits and vice versa? Probably so. And if we look at some correlations among the leg traits themselves, the one that jumps out first of all is the 95, and that was the front hoof orientation and the front knee orientation. So the pigeon-toed, splay-footed with knock-kneed and bow-legged, pretty much the same thing, right? If that animal is going to be real splay-footed, they're probably pretty knock-kneed and vice versa. So that's probably something that can be combined. Also, we see that knee orientation and hoof orientation is somewhat correlated with the side view of that front leg, which is not necessarily surprising probably, but in this case, the direction is that, let's see, the bow-legged was more associated with being buck-kneed, and knock-kneed was more associated with being calf-kneed, and with a pretty high correlation. Rear leg, sort of similarly, where you've got the rear leg, if they were posty-legged, they were more bow-legged and pigeon-toed, and if they were more sickle-hocked, they were more knock-kneed and splay-footed. So we've got some relatively high genetic correlations here, so these traits could be used to predict each other. But again, there were lots of zeros. And so if I didn't have it on here, there's lots of these correlations that really were not significant. Some of the other correlations that came up to be fairly high, body condition score was actually highly correlated with some of our feet and leg traits. And so the directions here, thin cows were associated with more open divergent claws, they were small-footed, they were more bow-legged and more pigeon-toed. And more fleshy cows were the opposite. They were scissor-toed, more likely to be scissor-toed, big-footed, knock-kneed, and splay-footed. And then remember, the composite score was what the evaluators scored the cattle just as an overall measure of soundness if they had to give them just one score. And the traits that really came up as highly correlated to that were the foot traits, so front and rear hoof angle and hoof depth, and then the side view traits, front and leg side view. So it appears that an evaluator tends to be highly influenced by those traits. Those are the ones that are most significant. So then the next step was to look at how do these soundness traits correlate with or impact our production EPDs. And so again, we'd love to throw this all into one big model and let it work, but this is a lot of traits and not a ton of data. So we grouped them into front limb, rear limb, and then a few others that wouldn't fit, and then correlated that with our production traits. And so this came from the Red Angus Association, both phenotypes and EPDs for the normal production traits, plus the two indices that they calculate. And so these would be genetic correlations between the in this case, front limb traits and the production EPDs. And so some of the interesting things that came up here, and none of them are high, right? We wouldn't expect there to be one thing we can measure on feet and legs that's going to predict 80% of weaning weight or something, right? We know that's not going to be the case. We would expect them to be low. But some of them that came up as kind of interesting is the front hoof angle and heel depth are the first two here. And in general, what we see is that a more sloping hoof or a lower heel is associated with a little bit lower growth, a little bit lower birth weight, weaning weight, yearling weight, a little bit lower heifer pregnancy, that's over here, and a little bit more calving knees, maternal. Front leg side view is this one here, so that's looking at the front leg from the side. In this case, more calf-kneed cattle tended to be a little higher growth, a little more yearling weight, a little more weaning weight here. And a little bit higher on some of the indices, the grid maker and the stability EPD and the herd and grid maker indexes. What's also kind of interesting, this is their maintenance energy EPD, so an estimate of how efficient those cows are. And we see some correlations here that were pretty significant, not high, but significant. But in general, lower maintenance energy, which is good, the lower number is good for that EPD, lower maintenance energy was associated with the more upright hoof, taller heeled cattle that were a little more calf-kneed and knock-kneed as opposed to the opposite. Again, low, but interesting correlations. This is front hoof orientation, so knee and hoof, so the pigeon-toed, splay-footed. Those are associated with a little bit higher weaning weight, but actually a little bit lower on the stayability and the herd builder index, which herd builder has a large component of stayability. The composite scores were associated with a little bit higher weaning weight, but a little bit lower on the indices. So those were interesting. If we look at rear limb, there was not as much that came up as being sort of larger correlations here, lots of sort of 0.0s. The ones that came up a little bit higher were that rear claw shape and rear leg side view were associated, so more scissor-clawed animals were associated with lower index values, lower stayability, and lower herd builder. In addition, those composite scores were actually associated with a little bit lower. So then the few others, the last few structure traits and production EPDs. Body condition score, not really a structure trait, but we analyzed it. And as you would expect, that came up as being a little bit significant with some traits. So higher body condition scores is associated with higher weight traits. That would be sort of expected. But a little bit interestingly, higher body condition score cattle had a little bit lower heifer pregnancy, a little bit lower stayability, a little bit lower on the herd builder index, which was a little bit interesting. These are the front claw shape. So scissor claws were associated with a little bit lower birth weight, but higher growth in weaning and yearling. A little bit higher metabolizable maintenance energy as well. And foot size, as expected, bigger footed cattle were just bigger. that would be expected. And then our composite scores were associated with higher weaning weights, yearling weights, and higher on the indexes as well. Just real quickly, one thing we looked at was if we took our scoring system from the 0 to 100 and changed it to a 0 to 10 or a 1 to 10, so transforming it to a little bit simpler system, we lose a little bit of power, but not much. Heritabilities go down just a little bit, but they don't change too much. So probably a 1 to 10 would be a good proxy for the 1 to 100 and quite a bit simpler. So what are some takeaways? We know, and the literature would support, that feet and leg traits, structure traits have low up to maybe some moderate heritabilities, and so we can do genetic selection and we can make genetic progress. We can probably simplify and do a 1 to 9 scoring system, and that would probably be fine. We probably don't need 13 different structure traits, that there's probably, with the correlations between those, we can probably find a little bit simpler system that would still capture a lot of the variance that we see in cattle. And we do definitely see some low genetic correlations that would indicate that some of these traits might be useful to indicate other traits, and obviously stability is the one that we really thinking we can maybe get a little bit of information about. If we can predict which cows will stay in the herd longer as yearlings, that's really the goal, to make those predictions better. Obviously, no geneticist ever ends a talk without saying we need more data. And so we definitely need more records. On lowly heritable traits, you need a lot of data. And that's something that Lane has been working furiously on over the last year, is getting more Simmental records. And I believe you said we're up to about 1,500 records, Simmental records now. And so we're working on that analysis, kind of the same type of analysis, looking and seeing what the heritabilities are, and maybe more importantly, what the correlations are in the Simmental data, to figure out what types of things we need to go forward to collect to make good predictions. So with that, I'll let Lane answer any questions that you might have as we take a little break and switch speakers again. So, any questions? And I think Megan is up, right? Okay, fantastic. And again, Lane and I will be around, so if you have questions, we can for sure talk about that later.
But glad to be back here to talk to you again about another project that we're just getting started at K-State. This one is centered around measurement of methane production and greenhouse gas emissions in grazing beef cattle, which is a little bit of a different twist than a lot of what's out there being worked on at this point in time. So just to give you a little bit of background on greenhouse gases are, of course, gases that trap heat within the atmosphere. And if we look at US GHG emissions, what we see is that carbon dioxide is really the largest contributor to that. But what you notice there is that methane makes up about 10% of US GHG emissions. And methane gets a lot of attention. And one of the reasons that it does is that it's 25 times more potent than carbon dioxide in terms of its global warming potential. And of course, methane is produced during fermentation in the rumen, and it is mostly emitted from the mouth. So we do have a little bit coming out of this end and a little bit coming from manure, but most of that methane that is produced in the beef value chain is going to be coming from enteric methane, which comes out of the mouth. So if we take a look at where emissions are generated within beef production, approximately 80% of greenhouse gas emissions produced per unit of beef, in the US at least, occur basically in the pre-farm gate. So basically everything we do in producing the animals, the other 20% comes from processing and transportation of those products and everything else that comes downstream. So if we look at that 80%, the cow-calf sector is the largest contributor to that 80%. And that sort of makes sense when we think about it. So this is cow-calf sector compared to stockers and feedlots. And that sort of makes sense. So 63 to 80% approximately of GHG emissions come from the cow-calf sector. So that's about 60 to 65% of the total from the industry. And that sort of makes sense when we think about it, right? Because we have to maintain cow and bull inventories year-round to produce one saleable calf per year, right? That's producing that beef product. Cattle have a long life cycle compared to other protein sources like chickens and pigs and things like that, right? And there's not a lot we can really do about that piece. And when cattle are eating forage diets, they produce more emissions than they do when they're eating concentrate diets. And of course, in the cow-calf sector, we're often grazing grass, right? But nevertheless, despite all of these facts and figures, beef cattle production is really only responsible for less than 2% of US greenhouse gas emissions, at least in 2013. So the major contributors are, of course, transport and generation of electricity, right? So I would probably argue that this isn't maybe a massive problem in an objective sense, but we do still have room for improvement, and sustainability is, of course, about room for improvement, and we want to provide the same human nutritional value of food with lower emissions for greater environmental sustainability, right? However, despite the fact that this is less than 2% of US emissions, I'm going to argue this is something that we still need to think about as an industry because for two main reasons. One is, as I'm sure you've noticed, this gets a massive amount of attention in the news media, and we hear all sorts of stuff about how eating meat, and especially beef, is bad for the environment, right? So that's one reason to care about improving emissions. But also, and maybe more importantly to this crowd, there's also an economic reason, right? So basically, methane production is essentially loss of the gross energy value of feed, okay? So whenever we produce methane, we lose energy that is not being able to convert it into something that the animal can use. So this contributes to feed efficiency of our industry, right? So basically, cattle produce volatile fatty acids in the rumen, right? And those VFAs are greater than 70% of the energy source for ruminant animals. So they're kind of a big deal, right? And the three VFAs that are typically produced are acetate, propionate, and butyrate, okay? So for cattle that are being fed on forage, there's a 70-20-10 ratio for acetate, propionate, and butyrate. Now, when we switch animals to a concentrate diet, that ratio changes to 50-40-10, okay? That is important because the butyrate and acetate have extra carbons that are produced during the production of those two VFAs that can be used to produce methane. So the more acetate and butyrate that we are producing, the more potential we have for producing methane, which is why you see higher levels of methane emissions on forage diets as compared to concentrate diets, okay? And so the energy that we lose to methane production is generally about 6 to 7.5% of energy intake, okay? So that's a lot of money that's basically going up in smoke, as you will, whenever we produce methane. And they're ruminants, right? We're probably going to produce a little bit of methane because we're utilizing forage resources, but if we can reduce the amount of that energy that's lost to methane production, that's more energy that's going into beef cattle production, right? So what can we do? If I'm allowed to paraphrase Temple Grandin a little bit, we can't manage what we can't measure, and as an animal breeder, I'm going to say we can't improve what we can't measure, right? So this is a big challenge for a lot of important traits in the beef industry. So disease resistance is a great example. Forage intake is another example, right? Really tough to measure direct forage intake of cattle on pasture, right? Maintenance energy is another trait. And of course, fertility that we talked about this morning is often another challenge. But I'm going to argue that we are to a point with technology now that we can measure methane production with some of the new technologies that exist, even on grazing animals. And I think that's the key, because that's really important to the cow-calf sector of our industry, and that's where the majority of those emissions are coming from. So before, what you'd have to do is you've got these ventilated head box systems, right? Or they might be in an entire chamber, and that would capture total methane emissions. These will just capture enteric methane, but that's the largest portion. So you'd have to have animals that are halter broke or very, very calm to put them in these basically enclosed boxes and then tie a little thing around their necks so that none of the gases get out back this way. And you have to feed them all manually and water them manually and stuff like that. As you might imagine, this is not very fun to do on a large scale and is probably impossible to do on a large scale. But I would argue with technology, we can now do some different things to capture that data in different ways. So one of those ways is the green feed system. And we're going to be purchasing one of these units here in the near future. But basically, this little box right here basically takes the place of that head box, right? So it's not enclosed. We can take animals that are grazing out in pasture, if we put this thing on a trailer with an energy source, and we can collect that data for animals that are grazing out on pasture, where we really want to collect this kind of data, where it's really important. So for us at K-State, one of the things we're really excited about is we have a fairly new Incentech system, which can collect feed intake data and water intake data. So if we pair that with the ability to collect methane emissions data, that gives us a really powerful tool, we think, to address issues like feed efficiency and energy use. So that's one of the things that we will be pairing and measuring with some of our purebred unit, cattle at the purebred unit. So mature beef cows as well as growing heifers. So we think this is really interesting for a variety of reasons. So one is that there's really not a lot of data out there with gas flux measures on large numbers of cattle that are on grazing pasture. Second is there are really no genetic or genomic studies of methane production for grazing cattle that have been performed in the US. And finally, there's really no genetic evaluation system in the US that incorporates methane production phenotypes to produce EPDs for beef producers. And where I think this may really have a lot of value is in where we're looking at feed intake and feed efficiency within the industry. So just to give you a little bit of background on sort of how that system works, right? Because as you might imagine, a little box on a trailer, cows may be a little bit curious, but they're probably not going to go stand in that box going, what is this thing, right? So we have to have some way to get them in to utilize that system, and the way we do that is with bait. So pelleted feed can be dispersed at random intervals throughout the day. So whenever the animal's around, we can have that feed dispersed, and that encourages the animal to stick its head in there and hang out while it eats that feed long enough to measure the gases going in and out of that animal. And you can see here, this is just some sample data from green feed, but this has dates. You can see there's some variation in the amount of methane that's being produced per day, but we also have variation in the animals, right? So we have some that are producing a lot of methane versus some that are producing much less on a daily basis. So if we sample that over a long period of time, we should be able to get really high quality data on average methane emissions for those animals. So that's the idea. And what we want to use that data for is to accomplish a couple of different goals. One is the first thing we need to do is figure out how long a period of time do we need to collect this data. We had to do that with feed intake testing as well to figure out how much time and how many measures we actually have to have on those animals to collect accurate data. Second is we really want to evaluate the stage of production and how that affects methane production. So whether the animals are growing animals, gestating, lactating, et cetera. We also want to estimate the relationships between production traits. You may be noticing a theme from the fertility talk this morning, but we want to understand how methane production is related not only to feed intake, but to other economically relevant traits. So if we change that trait, what is the economic impact of that on other traits that are important to profitability? And finally, we want to develop a genetic evaluation for methane production and see how that really fits with feed intake and feed efficiency in particular. So that's all I've got on that. And I think Jenny was going to come up and do another quick presentation here on a novel trait. So I will turn the podium over to her.
In this section, we're kind of just highlighting a few of these interesting, novel, new things that potentially might have some value as we go forward. And so I'm going to talk very briefly about some work that actually Megan started at Oklahoma State and is continuing here. Again, the grad students did the work, and we get to talk about that. But looking at water intake. And so we've heard a ton about feed intake over the last decade. But what about water? And if you think about water use in agriculture, it really depends on where you're at. You know, even within Kansas, in this part of the state, we don't worry too much about water. We've got more water than we know what to do with half the time. But if you travel not very far west of here, water becomes extremely limited extremely fast. And so as we go forward and think about sustainability, do we need to really, as an industry, think about our water use? And when you start looking at water use in beef cattle, it's not a very long look because there is literally nothing out there. We don't know anything about how much cattle drink and how it affects their performance. We just don't have any data. And so We know, or we at least have some idea, especially maybe some work in other species, that obviously water has something to do with how they grow, and obviously when there's heat stress, they need more water, but it's just not been looked at. And so there is, along with the feed intake technology that has come on in the last decade, many of these systems now actually have the ability to measure water intake on an individual animal, in the same way they do feed intake. Again, you look at the data, there's not much out there. There's a little bit in dairy cattle. And what I wanted to really emphasize here is the incredible amount of individual variation. And a lot of you guys that deal with cattle kind of know this, is that some cattle drink so much more than others. And obviously it depends on the day, but even in the same group, in some early work in dairy cattle, everything from 14 kilograms a day to over 170 kilograms of water a day for cows in the same group. Okay, enormous amounts of variation. One of the very few studies that's looked at beef cattle looked at a few different breeds and found there were some breed differences too. Different breeds of cattle drank on average more or less. But there has been, up until now, nothing genetic really to look at what's going on with water intake. Now there's been a little bit in mice, and so if you go to the literature, there's some mice work, but really nothing in beef cattle. And so that's really what the project set out to do. And so really briefly, there were 5 groups of cattle, feedlot cattle on feed at Oklahoma State, roughly 100 to 110 calves in each group, so a little over 500 in the whole data set. And again, I wanted just to draw your attention to the variability. And these were fed in different seasons, and you can probably figure out which was which, but anywhere from 25 to 50 kilograms of water per day on average. But look at the variability in those groups. The low animals are drinking 20 maybe kilograms a day, up to close to 100 in some cases. So incredible amounts of variability in these cattle. If you look at some of the relationships between water intake and other traits, we see that there, as we would expect, there does tend to be a relationship between water intake and feed intake, right? If they eat more, they drink more. That kind of makes sense. We would expect that. Genetically, when you look at this, these are the heritabilities for the traits that were measured on the study. Lots of traits that we would measure otherwise, daily gain, RFI, things like that. But water intake was up around 0.4, heritability of water intake. So meaning if we select cattle that can drink less water and still perform, we can make progress in that. Again, correlations, dry matter intake and water intake, fairly moderately correlated. If they eat more, they're going to drink more. But what's really interesting is gain. Almost no correlation, and high standard errors, but no correlation, meaning whether those cattle were gaining a lot or on the lower end of gain didn't really seem to be related to how much water they drank. So that's something interesting as well. What about carcass traits? Just kind of plotting out the water intake versus the carcass traits, and what you see are a lot of scatter plots, which is a good thing. What this is telling us is there doesn't seem to be much relationship between water and carcass. If you look at the genetic relationships, very low, except for maybe carcass weight. Cattle that are bigger drink more. That's fairly expected. But otherwise, what this says that is if we choose to select for decreased water intake, not really going to affect gain from the previous slide, not really going to affect our carcass traits. And so that's a good thing as well. And so again, just something to think about going forward, depending on what area of the country you're living in. Is water something we need to think about? We know that we can measure water as we measure feed intake for cattle on feed in an Instantech or GrowSafe. We know that we've got a ton of individual animal variation that we could select on that. Seems to be relatively low relationships between water intake and other traits that we care about, and of course there's a lot more work to do here, but it could be that we could select for animals that would use less water and not really affect our other production traits. And so going forward as an industry, we need to think about if this is something that we want to investigate further. And this work is continuing here as well, and we're collecting more data and analyzing more data to really try to understand those relationships and what happens with animals that are low versus high water intake. So with that, that's another little thing we're working on. I think Bob is here somewhere, paging Dr. Weber. Oh, there. You want to do questions first? Okay. For any of the, about any of the talks, any questions? Yeah.
So does anyone have any questions for Megan, Bob, or Jenny? Oh, okay, Ken?
Come on, Ken.
We should view this differently now, right? He's a colleague, not our boss.
That's true. That's true.
So, actually, is Megan around? My question is kind of targeted at Megan, but anyway, you guys can address this. But my question really has to do with this topic of bull fertility. And it has to do with maybe another way of trying to get at that. And it seems to me now with our ability to do parentage on commercial calves on a more inexpensive basis than what we probably were able to do at one point in time, we should have lots of environments where we have relatively large multi-bull breeding environments where we could actually genetically evaluate what I would call the ability to cause conception in cows. And perhaps it's a more direct measure of bull fertility than perhaps a lot of these other kind of attempts to get at it through maybe semen analysis or a variety of other things. So I don't know if I see Megan's coming up, and I don't know if she heard my question, or maybe one of you guys want to take that on. So Jenny or Bob or Megan.
I guess one thing that struck me, and it's probably been 3 or 4 years ago, we started hearing about some of the data sets where they would, on big groups of commercial cows, go back and say, well, look, one of the bulls sired, 60% of the calves, and there's another bull in the group, he only had like 2 calves or something, and we really need to select against those. But then I got to thinking, maybe it's not a breeding issue, maybe it's a fighting issue. I mean, do we want to select bulls that breed all the cows just because they're aggressive and they fight off the other bulls? Maybe that bull that sired 2 cows in a different group with a different set of bulls, or maybe if he's in a single sire pasture, maybe he's the best fertility bull out there. And I don't know how, in that set of data, how we could separate those things. Is kind of one of the thoughts I had about that one. I don't know what you guys want to.
Ken, I like the idea. The challenge, I think, is it's the combination of both fertility per se, so it's a semen quality and so forth, but also behavior and libido and social structure. And so I don't disagree that I think it's a good opportunity to collect data, because ultimately bulls that get cows bred at the commercial level is what we want, right? So, having the one that gets out and settles cows versus, laid up under a tree. I mean, the most fertile bull in the world, if he doesn't go mate cows, doesn't get any calves bred. So good observation. And I think ultimately, if we can capture data from commercial herds, would be a really interesting one to look at.
So I missed the first part of that, but, and Maddie, you can correct me if I'm wrong, but I think on some of the stud data that we do have some libido scores. So there may be some of that we can address, but I do think it's important, but I don't know that we probably have the data to address that at this point in time. But interesting question.
Hey, Bert. Oh, I think there's a microphone right behind you. Oh, wait, could you hold on for a mic, please?
Have you done correlations between these fertilities and sheath scores? Because bad sheaths are a pet peeve of mine.
So, Bert, did you say shoot score?
Sheath.
Oh, sheath score. Yeah, good question.
We have not done that. We don't have sheath scores on these, but if that is something that ASA has interest in collecting along with some BSE data, that's something we could certainly put in an analysis. We don't currently have the data to do that.
Lauren, one over on the right.
Willie has Willie's.
Question for Dr. Weber, and this has relating to fertility and gestation. We used to measure gestation length, and we're in a breed that has a longer gestation length, generally speaking, but I think genetically we could shorten it up. I mean, there are differences, and there's nothing that affects postpartum length as much as gestation length. So we could get the data. In other words, if you ask for breeding date, you will get breeding date. Now, as with scores, in other words, why did we call other? If you ask for breeding dates, you might get 285 days from calving date, but you will also get true breeding information if you ask for it. In other words, whole herd reporting will give you true breeding date. Some of us think that gestation length is a very important economic trait. So you can shorten gestation or a postpartum interval by shortening your gestation length. So I think that would be a fertility trait that I think we're missing here because there is differences. So why don't we ask for it? I mean, you didn't address that in one of your considerations. I think it could be, should be.
Yeah, good point, Willie. And I think gestation length is obviously central to postpartum interval determination, no doubt. But the reason I don't sort of typically think about it from a reproductive standpoint as a track maybe that we ought to go down real aggressively is much shorter gestation sort of can decrease fitness of calves in some cases. If we think about particularly calving ease, so there are a number of breed organizations use gestation length as part of their calving ease genetic evaluation. And what we observe is shorter gestation calves typically have lighter birth weights and better calving ease. Well, we can think of several breeds that have extremely light calves because they're 2-week short gestation or 10-day short gestation. How many of y'all in the room live in North Dakota? Okay. What's a 50-pound January-born calf require in North Dakota? Shelter, right? Hypothermia is a real problem. So I sort of look at real light calves due to short gestation as a lethal genetic defect that we've selected for. Okay? And we had 2 years ago here in Kansas in January, right when we were starting to calve, some really cold weather. And if you're not on top of a 50-pounder when it hits the ground, even in the barn, they can really struggle. So I'm concerned that gestation length is part of this from the repro standpoint, but it has some real negative consequences if we go too far.
I'm going to counter that. I'm not worried about 50-pound calves in this breed. I'm worried about long gestation length and 110-pound calves in this breed.
Fair.
So in other words, let's reach the optimum, and the only way we can, you said it, the only way we can find out is with data. So measure gestation length, calving ease, and birth weights, and we'll determine what the optimum is. In other words, I understand small calves in cold winters, but I'm also understanding big calves with long gestation length and winters when they don't get up. So, but that's not what we're talking about here. It's fertility and gestation length. So get the data and then let's evaluate it and figure out what the sires that are causing the problem and maternal grandsires, because the cow is the incubator. She holds it too long.
Yeah, her response to that calf signal is really critical in that whole process. Yep, no doubt.
Thanks, Bob.
Yep, good point. And I don't know, Lauren, do you guys collect any gestation or breeding data routinely? So AI breeding dates on cows in whole herd?
You're asking the wrong person. That's like a kind of a Steve McGuire question.
McGuire question.
He's like kind of
He's the database guy.
the iCloud atmosphere, yeah.
Yeah, we'll channel him.
Maybe Wade, are you in here? Or maybe someone else, maybe Janine, you work a lot with the database, or Sheldon? I don't believe we do. I don't believe so.
Okay.
I know other breeds do, but I don't think we do.
I'll add that to my list, Willie. fair enough. Thanks.
Got a question over here, crew.
Well, I'd just like to follow up on the point that Ken Odie made about some bulls appear to be, anecdotally, we think in multi-sire pastures are breeding more cows than others. Now with the era of big data and inexpensive DNA sampling, we can determine this parentage, and so can our customers. And what's going to happen, and we've got some data, Ken, we've been collecting some, and let me just typically, in a pasture of 100 cows with 3 bulls, we'll see results and have over several years now. One bull sires 51 calves, another bull sires 39, and the other bull sires 10. Well, we're selling these bulls to our customers, and they're paying $5,000 a piece for them. That's going to become an issue. I think we need to look at, is that a heritable situation? Is it an aggressive fighting situation? So what? Our customers are going to say, I'm not going to spend $5,000 for a bull that only has 10 calves, and they're going to figure that out very soon.
So with some of that, I guess, when I've kind of talked about some of Allison Manningham's and Dan Drake's work that looked at that at UC Davis, I've kind of built some of that as a management tool. Because we're probably happy if our best bull produces 51 calves, right? But if you're me, it'll be the worst bull that you bought that produced the 51 calves, and the best only had 10, right? But there may be things we can do with genotyping strategies to identify those bulls that are underperforming relative to what we expect, and maybe manage either how we're using them or remove them from those situations if they're not performing well, or maybe we want to remove the high-performing bull if he's our worst bull and put him in a different situation. So I've sort of discussed that as a management tool in the past.
So I know Ken has some awareness. I think maybe you're thinking of the same data I am, Garth Boyd's work at CSU.
Oh, hey, Ken, could you hold on, please, so we can get a mic on you?
My response to that is maybe the bull that sires 51 calves is the best bull because he sires 51 calves. Okay, thank you.
I think there is some interesting data, though, about sort of the social structure, right, and bull dominance, and it would be interesting to know how much of that is heritable versus a function of, well, he's just the biggest, baddest bull in the pen, and so he takes all the cows. So there's some complexities in there, and I've wondered about that before. And some of the obvious solutions are either reassort the bulls, but then by the time you figure out the new assortment, it takes you 2 or 3 years to figure out what the dominant bull is, and then he's gone. So there's some practical but not unimportant consequences of figuring that out. So.
Okay, so we have time for just one more question. I think Riley has someone. Okay, it's Gordon.
It's Gordon.
Question.
All right, there it comes. I think it's for Megan, and Megan, I'm going to go out on a limb just a little bit here. This wasn't directly covered, but it's directly related. And this is, or today is, presentations for producer education, producer help. There's recently been a change in BSE scoring where the veterinarians have removed droplets, no longer qualified as a secondary. As a matter of fact, they're not even doing primary and secondary abnormalities anymore. What is, and sure, not trying to put you on the spot, we need some help. I've already been hit with the question by our veterinarian, and it's asked, do you want us to do the BSEs on your bulls this year by the new standards, or are you more comfortable if we stay with the old way, because it is going to make a difference? And with us with fall calving cattle and coming up on BSEs here in a matter of weeks, it's already hit us. 80% of the population sitting in here they're going to probably get this question coming up about January or February. Any help you can give us there or opinion would sure be helpful.
Oh yeah, we're early in the process for an opinion on that, but I guess I'm going to go with Jenny's line, and more data is always better, right? So if we have data on any of those types of traits, there's a potential that we can utilize it to do something. If we sort of remove those from the evaluation, we no longer have the ability to sort of aggregate that and do anything with it to even know whether it's important. And that's kind of at the step where we're at now, is figuring out which of those traits are the most critical in sort of pregnancy success. And that's something we're going to be looking at moving forward. I don't know that I have an answer specific with data to back it up right now, but my default would be that more data is always better until we can determine that we don't necessarily need that data to do a good job at looking at those traits.
Yeah, I'd sort of echo Megan's comment, and principally from the view of, as bull sellers, getting bulls over a breeding soundness exam is pretty important hurdle, right? The challenge we have in many cases is, well, we'll talk about our purebred unit here. You guys visited yesterday up by Stanley Stout. We sell bulls in early March. Well, for most of our commercial customers, they're not going to turn bulls out for another 60 to 90 days. So how important is it to make sure a bull passes a breeding soundness exam 90 days when we know there's potential for substantial weather, all kinds of other stuff? So that breeding soundness exam becomes really important as a seed stock seller, but I would argue substantially less important to the buyer, because there's so much time in between, and particularly for proximal drop. The relationship of that trait over time on yearling bulls, it declines pretty dramatically, at least our experience, even from January to early or mid-March in our yearling bulls. So I think part of the response from the veterinarian community is, well, if we know the particular trait is going to change a lot and has little relationship at the time collected to subsequent fertility and settling capacity of cows, why collect it? Well, I argue we collect it from a genetic improvement standpoint, because the more bulls that pass, the better off we are. It's an ERT to us as bull sellers. That's kind of my take. I would encourage you, if you can continue to collect it, do for a while until we can kind of sort out some of the genetic relationships. So.
Okay, I'd like to thank Bob and Jenny and Megan. I told you that they're an awesome, relatively young group of beef cattle geneticists. And I really am pleased that you took one of your Saturdays to come speak to everyone at ASA. So let's give them a big hand.